Just going to welcome you all whilst I'm still admitting people in from the lobby. Hi, everybody. Welcome to our latest webinar. Today we're going to be talking about ASRgwas, which is the latest in our ASReml-R tools, supplementary to ASReml-R. As I say, for those that have just arrived, do feel free to say hi in the chat. Let us know where you're joining from. If you want to tell us a little bit about what you're hoping to get from today, please do go ahead. As you know, the meeting is going to be recorded. We will share this meeting with you after the event. Do feel free to pass that on to colleagues or interested parties. More than happy for you to do that. If you have questions during the webinar, if you can post these in the chat, we'll try and get to them either during the event or at the end of the presentation. If there's a lot of questions and we're unable to reply, we will try and get back to you via email, or we'll put something in the follow-up email that we're going to be sending out. Yep, so I think that's it. And without further ado, I will hand you over to Salvador Gezan, who is joining us in the UK today, the snowy UK, very cold, very snowy here at the moment. Over to you, Salvador.
Well, hello. Can you hear me well? Okay. So I'm just going to share my screen, and I should say good morning, good afternoon, or good night, depending on where you are. And thank you for joining us. I imagine you can see my screen. I'm assuming it is the case. So I'm going to talk a little bit about the library ASRgwas and put it into the context of the GWAS, but also I'm going to talk a little bit about the concept and the idea behind the GWAS and why we decided to have a more flexible GWAS, and also a tool that it's more realistic with the data we have, and that can be made much more easy and expedite your analysis with GWAS. So we are all familiar with a little bit with the GWAS concept, the association studies, where what we do is we have a group of markers, a marker matrix, which can run into the thousands or millions, and we want to try to associate those markers with some phenotypic response. And we obviously, the natural way to think about this, there has to be a correlation between the state of the markers or the alleles in a given marker and then the phenotypic response. So hopefully it will be as easy as that, but it's not really. So we usually have our data from breeding trials or some sort of structured data. And obviously we need to play with different matrix and with different elements to kind of control for the potential structure, for the family structure, and maybe even some other design elements. All of this is very standard in most of the GWAS software. And I'm going to split the analysis on the steps when we do this GWAS into three parts: the phenotypic data, the molecular data, and the proper model fit. In the phenotypic data, normally what we start doing is looking at the data that we have from our response, and we try to assess the quality of that data that we might have. So this implies if we have a pedigree, we check the pedigree and we can calculate a heritability. If we have a genomic matrix, we calculate the genomic matrix and we also calculate a heritability. We obviously want a heritability that is positive, reasonable in order to proceed. We look for outliers, we look for other markers, maybe for covariables that we need to add, and something like that really tells us we have a bit of a more clean data set. Often in many of the GWAS software and also in the ASRgwas, but it's a bit more flexible, but sometimes you need a given response, a single response for each of the genotypes that you have genotyped. So in that case, what I call the adjusted mean, we try to fit the original model with blues, and we obtain a single value that represents the mean, in one sense, or the best predicted value of that individual for the experiment. And those are the typical cases on plant breeding, where you have incomplete blocks, row columns, or some spatial components. And at the end, you get a single value for each of the genotypes with the weights or without the weights. And the weights will be important if you're doing a two-step analysis, and we can, again, incorporate those weights into the second part of the analysis, or you just ignore the weight. The other step is the molecular data. Obviously, we might have a very large matrix with probably lots of missing values that needs to be processed. We have to do the QC or the quality control. We eliminate markers, we eliminate some individuals, and we obtain the two main matrices, which are the genomic matrix, which I'm calling here the GA, or sometimes called kinship matrix or K matrix. That's the one that is going to allow me to control for family structure. And also we obtain a Q matrix, which is a structure of the population. This is done usually using the marker data that we already filter, or the kinship matrix, and we obtain some eigenvectors to use on the posterior analysis. I'll detail a little bit more about the models in a sec. And then we proceed to pull those matrices together with the data, and we fit our model, and we obviously obtain some goodness of it statistics associated with the markers. So we find the markers that are significant, and we kind of explore the markers. Some software allow you to do some backward selection or backward elimination, so you can eliminate some of the other markers that might be a bit too redundant or too close. We have graphical output and so on for this model fit. And then obviously we have the luxury to do some validation. We will proceed to do some proper true validation ideally, or some form of cross-validation. So just a little bit of the molecular data, which is probably the most important bit. Obviously, we have thousands or millions, so it depends on the platform that you're using. We always need to do some quality control, eliminate markers that are obviously with too many missing individuals that have many missing observations. Sometimes we look for a high level of heterozygosity. Anything really, I mean, GWAS, we tend to have a lot of markers, so we pre-select them. In some cases, you can perform some form of pruning to reduce the number of markers, and that might be relevant if you're a bit concerned with the multiple testing, where you obviously don't need two markers that are identical describing exactly the same position or the same gene. So there is some work that could be done with the pruning. And then we have missing values. The missing values in this particular case, if there are not too many, you can do a simple imputation, or if you have more missing values, you can use some more sophisticated software. And I mentioned the imputation because in some in some human studies, sometimes they have a chip from 6 million SNPs, and they impute it to 12 million SNPs, and they do both of the GWAS, and they make a comparison of what they found. So imputation has quite a important role to play here if you want to use imputed data. Obviously, we obtain the kinship matrix, and we have the band-rated method or other methods to obtain the kinship matrix, and lots of software that can do it. And we obtain the Q matrix of our structure, usually by some principal components where we extract the eigenvectors, but there's other software such as Structure that will do that. So let me just sit a little bit on the model, and this model, I made it a bit more complex to add lots of fixed and random effects. But the first line really is just a GWAS, so it's a GBLAP model that we fit to do genomic selection. So we have a response variable, an overall mean. Then we have a series of beta effects, which are your fixed effects of replicate, if you have replicates or some covariables or whatever you need to use, you will put it as a fixed effect here. And then you might have a set of random effects, depending on the area where you are. This could be an incomplete block, could be a plot, could be a pen, or anything that kind of like defines a bit of that structure on your data. And then we have the additive effects, or our GEVVs, when we fit this GBLAP model. And obviously this is associated with a genomic matrix, which is our GA, and we obtain a variance of additive effects. We can have several fixed effects, we can have several random effects, and so on. And we can have the errors, which are normally assumed to be normally distributed with equal variance. But when we move into the GWAS, which is actually sort of like an extension, what we do is we have exactly the same terms, but we have now this term added, which is the vector SI from your marker data. And this is the one that is coded 0, 1, 2, because we count the number of alleles on a reference allele. So it's going to be like a continuous variable, and then we have an alpha, which is the slope, or in this case, the effect associated with each of those SNPs. And we're going to run this model from I equals 1 to all p markers that we have. We have the same fixed effects, and we may choose other set of random effects that we might choose. And here I describe the Q, or the structure associated with these lambda, which are fixed effect associated with those eigenvalues on the Q matrix. And then we have the CA, which no longer is going to be breeding values, but it's close to breeding values. This is associated with the family structure, again, if you have individuals that are related. And this, again, I like to think that this represents the unexplained portion of the genetics that is being left out from that marker that is obviously not explaining everything that happens with those breeding values. So that is our model. It has a lot of more extensions, and you see different software allowing you for maybe some fixed or maybe some random. Here we put everything. So you might have used any of these packages. Tassel is a specific package for this. It runs very well. RBLAP has a nice routine to run an RBLAP model for your GWAS. It calculates the genomic matrix and so on. I find it a bit limited in the number of effects that you can add. GAPIT is another one also that is very widely used. And then we have the StatGen GWAS. All of them have properties and things that do some or other things a bit easier or more stable or so on. So what we decided to do is we decided to build a new package that has uses ASReml in the background, ASReml-R. So all the elements that we can specify in ASReml, we incorporated into our ASRgwas library. So we can consider lots of random effects, fixed effects, and heterogeneity, and also not only link it to one observation of one genotype. We can have multiple repeated measurements or repeated observations of the same genotype or clone or whatever it is. So we build this package, and this is what I want to show you, that it's integrated. So it calls ASReml in the background, but it's more than that, actually. Prepares everything for you. So the idea is that you put you have your data, and there's a first run on a function that we call PreGWAS. That what it does is looks at your data and say, oh no, you have some individuals that should be out because they have too many missing. It makes some decisions for you. You can specify some filtering, and you will eliminate a few things. But at the end, it prepares the matrices and makes, for example, the G matrix to have the same dimension of your M matrix, and so on. So it prepares all the things in order for you to be ready to do. And it gives a lot of reports, so that you might find, for example, that they say there's duplicates, and the duplicates create issues. You need to use stops there. You need to see what happens with those duplicates, eliminate one of them, or assess what happens with them, etc. Once that is prepared, and this is where we prepare some, we are ready to fit the GWAS model, where we have a specific function, very flexible, because you can provide what you got from the pre-GWAS, or you can even provide your own genomic matrices if you want to use a slightly different approach or want to check some other things, etc. And we have some functions that do the post-GWAS. Again, you can download it from here, or you can ask us by emails, or have some comments and so on. So there is lots of details on that library, and I'll come back a little bit into these details. But as I mentioned, it prepares the data, audits a little bit how's your data and what's going on, makes decisions for you. So this is where some people might be very happy to say, oh yeah, you made the decision for me, I don't need to worry about that, except if there's a warning. And some people might want to look back at those warnings and say, no, I want to recheck what happens here. So it gives you that option. It fits the model. It also does a backward selection if you want to do. And we have some graphical outputs that are some of the traditional graphical output plus a few more to look at what happens on your output. We try to make it as fast as possible. We use some parallelizations, but we also use some C routines for matrix operations using the traditional GWAS procedures, but also we allow for exact analysis, okay? We allow for any number of fixed and random effects. Because of that, we also allow for replicated data. And in addition that, we allow for marker information that it's missing. So most of the packages only accept marker information with no missing values because of the way they operate with the matrices. What we do is we have a procedure that we approximate some of these information, some of the matrices required for fitting each marker individually. And then also we expanded it because we have the ASRgwas embedded together with the ASReml. We expanded the GWAS analysis to have some binomial data. So let me just go directly to the demo. Too much talking for the first 15 minutes. So I imagine you can see my screen. If not, just let me know. So I'm going to use different data sets. This is 4 or 5 different examples using mostly the base model that I show you. I need ASReml, for which you will need obviously a license. And then you can download ASRgwas, and we have a library ASR data that has the data that I'm going to pull, and there's information there where you can collect it. And ASR Genomics, and some of you might know it, is the one that processes the molecular data also all behind the scenes. So we can work directly with those things, no problem. So I'm going to, there's a few more information here, and actually if any of you is interested on this code, and interested in the presentation, just we'll have it when we post the final video. So the first example, I'm going to take some data from aquaculture. This is 1,481 individuals from salmon, and they were evaluated for disease in a specific study. They call it a challenge experiment, and they were phenotyped for the mean yield score, so they provide a score, and an amyobic load, which is a quantity measured with the qPCR. That's the one I'm going to analyze. And obviously my objective is to find some markers that are associated with any of these diseases, but I'm going to focus on the second one. Now, all individuals were genotyped. I don't have too many missing values, but I have around 17,000 SNPs to analyze. So this is just to show you a bit of the description of the data and the reference. And this is my phenotypic data. It's very simple. I have one record for individual and a couple of phenotypic responses, and then I have the M matrix that actually it's here, coded as 0, 1, 2, with the names of the markers. Sometimes you have extended these names to have the main allele. In this case, I don't have it. And I'm calling this 1-to-1 because the dimension of my phenotypic data is 1481, and that's for the phenotypic data, and the dimension for the genomics data is 1481 also. If for ASRgwas, it doesn't need to be on the same order because it's going to check internally. Okay. So yes.
Does ASRgwas accept categorical traits such as disease score?
It's not extended to multinomial models. It's only extended to binomial models at the moment, but it shouldn't be that difficult. Now, in terms of a score, often what we use in this case is the mean yield score of several measurements of the same individual. So it kind of approximates to a normal distribution, but it's not exactly the same. But yeah, so it's not that extended, but it has the potential because we have that in ASReml-R. Okay, good question. So I'm just going to do a little bit of summary of my data, and then we can have a histogram. This is just the preliminary checks. Now, this is the main function, the PreGWAS, that I want to show. We have lots of things. So this is where you specify the phenotypic data, the individual, which column has your individual genotype name, the genomic data, what is your response variable, weights if you want to add weights, map if you have a map. If you don't have the map will be generated internally with just a dummy chromosome, a single chromosome and dummy positions. If you want to rename the markers in case you do some transformations, options for the Q matrix. And then if you have your own K matrix, you can provide it and it will be filtered and so on. If you want a K matrix or kinship matrix returned to you, because this PreGWAS generates just the inverse of that matrix. And there is a lot of more options in this ... because all the options that are in ASRgwas can be done here. So I'll show you a little bit how it goes with my pre-salmon data, which I'm going to run. And while we wait, I'll explain a little bit what happens with this one. This is my phenotypic summon data. ID is my column, my response variable. I requested the Q method to generate the Q matrix vectors, which I still haven't specified how many elements I want. The method for the kinship matrix is the Van Reyden, and all of these options here are options that we have in the ASR genomics. Filtering for minor allele frequency, marker call data, so proportion of missing marker by individual, if you have a threshold for heterozygosity or for inbreeding. And if we want an imputation, in this case, it did an imputation. Now, you don't need to do any of that. You can put it at 0, and then if you already prepare your matrix and so on. And then ASRgwas is going to report a lot of things. So let me just increase this a little bit. Obviously, it starts looking, and let me move this one a little bit to the side. So it starts looking at your marker data. If you eliminate some individuals, actually we have 1.46% missing SNPs, which were, in this particular case, they were imputed with just the mean. We have the geno data used for the Van Reyden, some information about the genomic matrix obtained, so some assessment diagnostics, if it has potentially duplicated individuals, if the range of the diagonal is too large, and so on. So this one will report things that are really suspicious, and it might try to fix some of them. Again, it's up to you to accept them or not. And then we take the inverse of that matrix, and notice that the inverse says that it was ill condition on matrix, so that's probably a determinant very close to 0. So we did a blending internally with a p.005, and then we have a matrix that is not ill condition anymore. So we are ready to kind of proceed. So this PreGWAS pretty much prepares everything, and you can see here we have the eigenvalues we need to use, a filtered genomic data, remember with the filter markers, so maybe filter individuals, a filtered kinship matrix inverse, a filtered dummy map data, your filter phenotypic data with everything you send, and we have the plot scree and the Q. So let me just show you a few things. This is just the dummy marker data, markers that were eliminated. Now we have 11,176 markers. And the scree plot is the one we use to decide how many dimensions we want to send for the Q matrix, and it's usually where this breaks, the elbow break. So I have 7 dimensions that I want to send, and that's what I recorded, this MPC equals to 7. And now that I'm happy with that, I can fit my ASRgwas. I'm just going to let it run because this takes a little bit of time, not excessive, but a little bit of time, and depending on which method you request also. So what I'm doing here, we have lots of options. So when you look into the help, you'll see lots of options, several random effects that you can add, fixed effects, a structure for your residual, weights if you want weights, the 2 different families. And then this is about memory usage. This is again about the dispersion on the binomial, and some things about your p-value threshold and the Bonferroni, if you want a Bonferroni, and the method to use, which is this P3D, and some other elements that we use to kind of update when we have missing values on the marker data. So there's a few more options, and together with the parallelization, if you want to use how many threads and things like that. So this model, which is running on the background, uses our genomics data, my response. Here I have the Q matrix that I obtained before, but I'm only telling it that to use the first 7 vectors, in this case, the first 7 columns, my genome data and my map data, and then I'm asking for a p-value, which is kind of a dummy p-value. This is for illustrative purposes. So I asked for a p-value that is relatively large. So humans use like 0.5 to the minus 6. And Bonferroni false unless you want. And the other thing that is important is that the P3D is true, which means that I'm using the approximated method where the variance components are estimated once, and then we do matrix algebra operations, fixing those variance components to obtain the slope of each of the markers. So it's relatively quick. And again, if you're using some form of parallelizations, which I'm not using here, the things will get a lot quicker. Okay.
There's a quick question just whilst I'm
yes.
running. And we've got a couple here, so let me go through. Somebody's mentioning that they tried the PreGWAS for data and noticed that it wasn't accepting marker scores for polyploids, i.e. 0, 1, 2, 3. But GWAS ASReml seems to have no problem with these marker scores. Is this something that we're aware of? Is it an issue? We can come back to it.
No, it's probably because we set up a restriction on the scores of the marker data. I mean, it's easy for us to modify that one. So if you let me know what more details later on, the person who asks more details, we can generate it and see. I mean, we put some restrictions because obviously we were thinking on diploids, and we want some of the users to not worry too much about the details. But those are important things to accept. And before you go into the question, the other question is this GWAS ASReml, because you kind of re-input everything, it allows you to, let's say, use another kinship matrix, manipulate your marker matrix. For example, you can have your genodata to be expanded to additive and dominant effects or some other parametrization of your marker data that you didn't have before. So we didn't want to pack it all together that you just click and it runs everything. It clicks, runs, gets some of the things, and then you can manipulate, fix, and even later on when you go to this GWAS ASReml, you can decide to trick it even more, again, for the advanced user. So what was the second question, Carrie?
The other one was, are we only set up for diploid data at the moment?
In theory, yes, we are because of the genomic matrix, but this section here allows to do whatever you want. So as I mentioned, you can replace this marker matrix by something that is not diploid. You will provide your kinship matrix inverse on something that is tetraploid. So it's and the same thing will go for the Q matrix. So is the PreGWAS the one that is a bit more restrictive? The GWAS ASReml allows you with pretty much anything. And as I mentioned, anything also with fixed effect, random effects, heterogeneous errors, and some other things. Okay.
Lovely. Let's continue. There's a couple more,
but
we'll pick them up after the presentation.
Okay, so it did run relatively quickly. It runs a base model and it gives me some output from the base model, but then it runs the marker information. It tells me how long it takes to go through the markers, really nothing. The base model is the one that takes a lot. And then the rest and some effects that it found and so on, and look at the fault discovery rate for the 9 markers that found. So this is a relatively high fault discovery rate. Again, this is dummy data. Well, it's real data, but I'm not going through all the strictness of the p-values or the Bonferroni. So we have several objects. I want to just mention that if you have a doubt of what is the specific model being fit, you can always use the call function from the object, in this case, the GWAS for someone. And you can see a little bit what is happening behind the scenes with the groups. If you have fixed or random effects, they will be showing up here, management of memory and so on. Some variance components. In this case, I have the genetic variance component additive, and then I have, which in this case is the related to the kinship, and then I have a residual term. If you want something for your model, the object from ASReml is still there. And then you can obviously assess your diagnostic plots and say, okay, I have some outliers. I have to come back and assess what happened there. And one thing we calculated is this heritability. Heritability based on variance component, which is the additive variance divided by everything else. In this case, is 0.25 with a given standard error, not bad. And a heritability based on the PEV, which is one definition that Brian Cullis has in some of his papers. And finally, the markers that are found to be significant with additional information. Chromosome, position, minor allele frequency, this is just as a reference. And then this is the effect, positive or negative, that you might have on that marker on this response, which is the homeovic load. Obviously, you need to help yourself interpret which are the allele, the favorite allele, and which one is the favorite allele, unfavorable, I should say. We don't have the coding for the allele reference on this marker, so we have to go back to the definition of the markers. Standard errors, C-values, P-values, which are not really that small in this particular example, and then an estimation of the variance explained by each of these markers based on all that information. So that looks really nice. We can get a QQ plot to again assess which, if the markers really stick out or there's a bit of a problem on the model, which in this case looks very, very nice because it kind of aligns very well. And then we have those extreme observations which don't align with the lack of significance. So, and then we can perform some backward selection. So I like this because then we have all these markers. I mean, some of them possibly are in this close to the same chromosome. They might be on the same area. So we might want to do something. And then again, you can do a Manhattan plot, which we also have the function Manhattan plot to assess that. So what we can do is if we can select those markers. So I'm just selecting the markers and going to my original M matrix and selecting the markers that were selected as significant, which is this M cell matrix, which is going to have only 9 rows. And then I can tell ASReml to rerun, which is this part that I have in the code. I have it commented out because I'm not going to run it because it takes like 5 minutes. It's very simple because you just say, this is my GWAS object, which is just the same I put before. This is my genomic data selected, or the subset of the markers I want to test into a backward selection. My reference variance components is the first one. This is just to calculate the variance explained. And heritability, if you calculate heritability, I put a value for p-value threshold of 1%. This is slightly different than this. Remember, I'm just assessing of potentially 9 markers, which of those are significant to be kept. So this is a backward selection. It puts anybody who is below that 0.001. The first one will be dropped, the model refit, drop the next, calculate the p-values again, drop the next one, and so on. And it repeats the process, and that's why it takes some time, especially if you put too many. And at the end will give me what is the set of significant markers. So I already run this, so I have it saved as an object, RDS. So I'm just going to show you, it's called fin. This is the model call, and you can see here out of the 9 markers, 1, 2, 3, 4, 5, 6 end up being significant. I can show you those who were significant. Now the effects are going to be a bit different because now all these markers are simultaneously onto the same model, not individually. So there's changes on the effects, changes on the standard errors. Remember, there might be collinearity between them. And then obviously changes on how much variance they explain. And the overall model in this case is telling me that these 6 markers are explaining 31.7% of the genetic variability based on the base model. So that's nice, and again, I think this is the way to go in the sense that after we find something significant, we put them all together into the model. This plot here is a marker plot, we call it. It just shows you a little bit of more information about your markers, the 6 ones that were selected, and you can see the states of 0, 1, and 2, and this one, for example, has a positive effect, which you see here on the first one, and then you have an increment. But in some cases, you will see here, for example, there's not much difference between 0 and 1, and 2 really seems to be like a drop. Again, eventually, we could test that with another specific test and so on, but we can assess if which of those states of the genotypes are the one that we want to see, and then what happens, what big the difference, and what happened with the outliers and so on. So this is not very, very clear in this population, but that helps, okay? So that's one example. Let me just clear this. I have an example where we have replicated data. In the case of replicated data, this is an apricot study, agricultural, where they actually took samples of 250, DNA samples of 251 genotypes. That's actually was the original number, but end up being 153 individuals cross between 2 cultivars. And we have 474 records. So we have a bit more than 3 times the records per individual. And this is because there were lots of observations that were taken. There was 3 lots of observations taken from each of those individuals and processed differently. So these lots represent blocks. So they got a few samples of the fruits and they process them by lots. And then we measure several things. And then obviously, so now we have 3 records, more or less, 3 records per plant or clone. So you can see here the genotype 2 has 3 records, and we are only using the data from one of the years, 2006. This is fruit weight, hue, ethylene, and other things, glucose, sucrose. I'm going to focus on sucrose, and you can see there is some variability. So one dirty way to do this is just to take the average of those three elements, maybe by a simple model and then lot as a fixed effect of replicate. But the other thing is to use directly all this data, and that's what we're going to do. So, so 474 records, and I have almost 61,000 markers to use. So, and
relevant to marker amounts. He's asking for big data, such as 500k individuals and 100 million SNPs. What sort of timing are we looking at to run the GWAS, or does it depend?
It does depend. I mean, how many individuals did you say was?
So we're looking at 500,000 individuals and in the region of 100 million SNPs.
It's going with that number of individuals, it's going to hard to manage everything in R. I will say you won't be able to do it in R because the matrices are too big, and the kinship matrix will be humongous also. So I will say it's going to be very difficult, and they have to devise approximation methods. Also, I'm not sure if everybody will have information. So if we're talking about a large number of individuals, are they all phenotype? If they are, then we want to use all of them. But if it's only a small portion, then we fit the GWAS in a small portion, and then we go to the molecular data of the rest. Now, in relationship to, let's say, 12 million or 50 million SNP, it's not a problem. I mean, R is going to struggle, but there's ways to, you can sample the matrix to calculate your kinship matrix, and then we can run this GWAS ASReml on everything, or we can run it in portions, even in different computers, and then pull all the data together if we have problems, for example, in R reading the full matrix of all the marker data. So there are tricks. Obviously, we need to sit down to see those tricks. Okay.
Great, thanks. Let's continue. We've got more questions, but
okay.
In the interest of time, we'll carry on.
Okay, and yeah, I have a couple of more examples. So and so again, this is just the PreGWAS on the same data, the band rating, some filtering for the data, and then I have my ASRgwas, which it runs very quickly this time because I mean, even if it's 40,000, well, the filter of the marker was only 10,000 at the end. So notice that we started with 60,000, and there's a lot of markers being dropped here because of the proportion of missing values too large. In this case, I use a more strict level, 0.4. You can change that and we can see how that goes. But anyway, so, and then I'm running my ASReml, the GWAS ASReml for sucrose. I'm using the kinship matrix. I'm using 5 dimensions for the Q matrix, same p-value and so on. Now, this is the important part. I'm using an extra fixed effect of lots, which has to be defined as a factor. And then I can define more random effects. So you can actually do a list. I can do this one like a list of factor 1 and then factor 2, whatever it is. So I'm just not going to process too much that, but there's many options for more markers. And also the residual, I'm asking for heterogeneous error by lot. So I have this suspicious that something happened. You can see the model. You will see again, the lots is a fixed effect. And then also when you look at the residual part, you will see it as a dsum by lots. You have a heritability estimated on that model, which is 0.79, which is quite nice, again, based on those 3 errors. And then we have a PEV heritability, which is also very good. Remember, that's the base model. Again, some residual plots to assess. We seem to have a couple of outliers here and there that we might want to do something. And we found 3 markers that happen to be significant from this lot of almost 10,000 markers. No problem at all. So you see, actually, one of the things when you look at this variance component, you have the genetic effect and you have the 3 error variances. And this is what I was mentioning, that because we did it with calling ASReml, we can have all the fixed effect, random effects. You don't have to worry about pre-processing your data to get a single response by genotype. The library will take care of where the replications of the genotypes are and so on, and define the factors properly. And so that's the whole idea. And eventually we can add more complicated structures or residuals, but I think with this you can have whatever fixed and random effect you might be interested, you know, pen effects and so on. A couple of the plots, this is a QQ plot. Again, we have, which seems to be a few markers, and the Manhattan plot, which I like to show, I didn't show before, but again, we have two markers that seem to be actually on the same position out of these three that I'm reporting here, which will be these two here, plus another one that seems to be something. Now, again, my p-values here are really relaxed, so I don't have much to do with that. But again, it's just to illustrate to you that we can do any model, which the way I will think, you can do any model with any complexity that you want, and why not? You don't have to reduce your data to a single value because of that's the restriction of the software. So now, next, I want to show, I have two more examples, and I want to show a binary response variable. And this is very, very common, particularly I've seen it in lots of like disease, and particularly in aquaculture. This is a data set from rainbow trout. And actually, I already ran the model because this model takes probably an hour or so, or maybe a bit more to run. Depending on the parallelization you're using or you're not using. Because what happens is that we're going to fit a binomial model, but we cannot use the trick of the P3D of the matrix operations. Every single marker has to enter into a model. The model has to be iterating, and then you get a estimation of the standard error, an estimation of the p-value, a calculation of the p-value, and then you store that one, and then you go to the next marker and you send. So it's very brute force because there is no shortcut with the way we do it with normal response variables. So that's why it's slow, but it will give you the proper standard error and the proper calculation under the proper distribution. So this data set corresponds to the IPN, viral disease infection pancreatic necrosis on rainbow trout. We have 749 individuals from this challenge experiment, and then we have a total of 40,000 markers. Now, one of the things is the response variable I'm going to focus is in mortality. And then this is coded as 0 when the fish is alive and 1 when the fish is dead. So that's important to know. So let me just load my output from the analysis I'm going to do. A couple of things, actually, the data has a lot of, let me just move this one a little bit to the side. The data has a lot of potential other factors that I could use. We have a family term, we have a sex, I have the ID of the individual, a contemporary group. We have also some potential covariables that we can use. So this is typical from all of these aquaculture studies, okay? So I'm going to consider sex and a covariable. So again, in this one, what I'm doing is I'm telling you how many individuals are in 1 on 0. So 271 individuals are dead and 478 from the lot are declared as 0. So no problem. So we have a good proportion. And then I can run my PreGWAS, which this is not going to take too much time. So we run the PreGWAS with the typical filtering. I don't have a map in this particular case, but I can request the Van rating and so on. And then I can proceed with some filtering of 0.2 or whatever you want to use. So I'm not going to focus too much on that, but this is just the typical report of markers that were eliminated. I have 0.42% missing. So that means I have a total of, what is the number, 39,000 markers to walk through. So this is why it takes quite a bit of time. So once this runs, you will see on your output that there's 25 significant markers with a full discovery rate of 79. Again, I'm using this 5 to the minus 4. So I'm not using a very strict threshold. The base model is here. And I don't know why it's taking a little bit to come up. There we are. Oh, because there's some things running. Oh, hopefully I didn't ask for anything. Okay, so this is the base model. It's using the binomial family with a logit link and dispersion of 1. You can modify the dispersion. And you can also use not only binary, but you can use binomial data. The dimension from the PCA from the Q is 3. We have the individual. We have some fixed effect of type weight, which actually is a covariable, and the fixed effect of sex, which actually is a factor, and I have family as a random effect on the model. And then homogeneous errors and so on. A calculation of the heritability, which you have to be very careful with this because this is based on the PEVs and it's hard to do calculations of heritability on binomial data. And then I have the variance components again associated with this GLMM with binomial data, which fixes the error to 1 because I'm saying the dispersion is equal to 1. And then finally, I can look at the markers that were selected. Now, I'm very happy with this because this is using the actual distribution of the response variable, GLMM. It's fitting every marker individually, which you can also do with the normal when you put P3D equals to false, which in this case, I think I put it here, P3D equals to false, because we want to fit everyone individually. And there's more tricks, so if you want to look into the manual, because there's ways to approximate that with missing data too, and so on, but there's no time here. Now, this is my significant markers. Some p-values seem quite interesting in this particular case. Again, the effect, now you have to be careful because that effect is on the logit scale. Okay, so that means there is obviously a logit transformation in between, so you can actually estimate the proper effect with predictions and so on. And there are standard errors and so on. So you have to take this with a pinch of salt because it's a GLMM. So that it's associated with this model that uses the binomial data. Okay. Carrie, it's a good moment for a question if you have a question somewhere.
Okay, let's have a look here.
Before I show the last example.
Sure. Okay, so Sanjeev's got a question about this particular apricot example with the F1 population. Can we do GWAS on F1 individuals? I will need more details on the questions later because I mean, obviously, if the F1 individuals, you have the parents' genotype. That's what I'm assuming. So you can reconstruct the genotyping of the individuals if the parents are inbred. And actually, we have a function in ASR genomics to do that, but you can do it with your own code. So you just have to create the molecular matrix of those individuals. That's how I'm interpreting. If I'm interpreting wrong the question, let me know later, and then I can expand a little bit more on it. Okay. Okay, Sanjeev, if you get back to us with a bit more information, or let us know if that's answered your question. Mo has asked, what does the workspace option do in the function?
The workspace will capture some of the RAM memory from ASReml to run the analysis. So sometimes ASReml doesn't capture enough memory and it tells you, it kicks you out. So you just say, please use more memory. I mean, you have to do a good management of the memory anyway. But good. Yep.
One more. Is there a predict function to predict GEBV of each individual using the effects of selected markers?
No, there is not a predict function that we have set up. I mean, we stop thinking about which are the markers that you have and which are the effects. But it shouldn't be that difficult. Now, that means you are pretty much predicting for the same individuals you have in this data. And our assumption is that normally you have another data set for which one you want to make predictions. So in that case, you will just take your effect and then apply to the other genomic matrix from, let's say, individuals from the current generation, not the past generation from where you fit the model. So we don't, but it's straightforward. Okay.
Another question, if we may, hopefully a quickie. Can we do, and how can we do rather, genotype imputation in R before we go to GWAS?
You can use, if I use normally a simple mean imputation when I have less than 5% missing. And then That seems to work fine. You can also not do the imputation, and the software will just fit each marker on the individual on where there is information on the M matrix. But I'd recommend if you do that, make sure you have enough a good filtering previously, so you don't want to end up with too few data for some markers. And then there is ways to communicate with Beagle, and there's other libraries to do imputation, but that's something that you probably want to do a bit outside of R. But I've used Beagle, connected to Beagle. But yeah.
Great,
okay.
Okay, just to say, for those that didn't join and are asking, yes, this is being recorded, and we will be sharing after the event. Also, any questions that don't get answered, we'll try and collate those and add them to the end of the email, and hopefully be able to answer those in the reply emails, in the follow-up emails. Thanks. Are we good to go, Salvador?
Yes, we're good to go. We don't have much time. I don't want to expand too much. So this is the last example, and this is an example that fits additive and dominance effect. So it's an extension of the GWAS model. I'm going to use the salmon data. Again, this is just for illustration purposes. Now, the way it works is that I have my genomic matrix that I use for my genetic or family background, which was based on additive effects by Van Reyden. But I'm also going to estimate another matrix that is by, in this case, the method SU, to estimate a dominance relationship matrix, which I'm going to fit on the model. Again, those two are just to model things on the background. And what I need to do is I need to reparameterize my M matrix that originally I had it as 0, 1, 2. Now I'm going to have that 0, 1, 2 for additive effects, and I'm going to make a copy of the same matrix that will have 0, 1, 0. So the 2's will be transformed into a 0, and that's going to be my M matrix of dominant effects. And so now I'm going to have two M matrices, one next to each other, on which I'm going to scan through all the things to see which markers are significant, and ideally find some markers that have some dominance effect. Again, you can use other parameterizations. If you have polyploids, I imagine sometimes you want to compare all homozygous against all heterozygous, or you can do some other things. I know there's different parameterization, dominance or codominance, etc. So I'm not going to get into that, but so the salmon data, I did process something in the background where we have the questions, because there's two things that I need for this. I obtain a pre-salmon A, which in reality, I obtain the filtering of the data and my marker data, and I obtain my Van Reyden kinship matrix. But I also did a pre-salmon D, and in this case, the only change I did is I have a SU method. There is a BT-SECA method and so on, which will give me a GD matrix. Okay, so their filterings might be different if you want to, but again, in this case, happen to be the same, which means the M matrix and the phenotypic data is going to be exactly the same. And here, what I'm going to do is just to have ASReml work fine, I'm going to copy my original ID that I have here. I'm going to rename it and call it ID for additive and ID for dominance, and you'll just see an extra copy of those two. I need this twice because I can assign the ID A to a kinship matrix of additive effects and ID D to a kinship matrix of dominance effects. And here I'm just doing some other little manipulations that if you have later on, you can check into the code. But one of the things I'm doing, I'm taking the MA matrix from the first part, the MD matrix, but the MD matrix needs to be renamed. The columns need to be renamed to have an extra something, so they are not the same, because otherwise you end up with the same covariable twice with different data. So you need to rename it. So I just added a D in front of them, D and an underscore. And then I also, with this line, I'm making a parameterization to my liking, which in this case means anything that is 2 is going to be a 0. And then I join the two matrices, and then you can see here how do they look. So this is just the added D and D. So now I have everything joined in a big, large M matrix that I'm calling MAD, and I proceed to fit my ASRgwas, which hopefully Yeah, I don't need to. Hopefully, it doesn't take too long, but this is going to take a little bit because it's a lot more markers to assess, double the markers, and they have double the matrices. So the key thing here was that when I specify genotype, now I don't say only IDA, I say IDA and IDD. And also when I say these are your kinship matrices, you need to specify for which term, which kinship matrix has assigned. For IDA has assigned this, and for IDD has assigned the other one, from summon A and from summon D. Principal component 7 as before, the molecular markers is AD, and the rest is the same, p-values is the same, and this is going to take a little bit of time to run, okay? So while we wait for that, which I still have a few minutes, and then we'll come back just towards the end, I just want to finish with the last few slides after this demo, because I have a few more elements to say. So one of them is the importance of having some form of validation of your model. You can think about, you can do a cross-validation. It's going to take time. And if you're familiar with the cross-validation and genomic selection, you leave some out, fit with the rest, and so on. In this case, you want to assess if they actually are going to finding the same markers that you think or not. So it will be a bit more tricky with a cross-validation. True validation is the ideal situation where you actually have a new independent or semi-independent study. This is what many journals are requesting, particularly in human genetics, that you repeat the same experiment to validate that your first study you don't have false positive, which happens also in the second study. So you end up with a common set of markers, ideally, between the two studies. So that's usually a bit difficult. So in breeding, what we normally do is we do some sort of like an operational check in our operational breeding. So we apply the selection to a group of individuals. They're still alive and they're going to be phenotyped. And once they're phenotyped, we can check if the marker that we selected originally really is telling me that the majority of the individuals that are resistant are the ones that have the allele, the favorite allele that we found on the previous GWAS study. So we do sort of like a proper operational validation of the data, and that's very common and good. So a few final words on the GWAS before I show you the results. So GWAS is now common use everywhere. I mean, if you're doing genomic prediction, you should do a GWAS. And the ideal of this package is don't waste too much time in doing a GWAS. If you did your filtering of data and so on, you can jump directly and get some extra from your box in this case, if you're using genomic prediction and you already have the phenotypic data, which, by the way, phenotypic data, again, is going to be very critical on all of this information. Do not forget that you need proper experimental designs, and because now this ASRgwas allows any structure, you can have a proper nice experiment with raw columns or whatever you want, different incomplete blocks, etc. Anything that you find is relevant for your analysis, it should be into a good experimental design. You'll need to manipulate your data, your molecular data particularly, and I mentioned this because some of the platforms have 40% missing, and you might want to decide if you're going to impute or how you're going to store it or what sort of filters you're going to apply. So again, all that is very important. And I have this mentioned here that if we are dealing with millions, and I have analyzed 3 million SNPs, I can open all the files at the same time, so you have to come up with little tricks and protocols and see how you do your GWAS. And in particular is usually you get a sample of markers to calculate your genomic matrices, and then you split your millions of SNPs into blocks that you process by blocks, maybe by 500,000 at the time, and then you have to pull all together the data before you run post-GWAS. A few last little things. The p-value issue is always going to be there, so you have to come up with how you want to decide which ones are significant. Depends again on the criteria, if it's an experimental study or not, or if something more definitive or you're verifying things. Some people prefer the full discovery rate, Bonferroni. I'm not going to get too much into that topic. I mentioned that you can extend the GWAS for non-additive effects and non-other types of dominance and epistasis, and there's many ways that you can expand that A matrix I mentioned something about the polyploids too. And I think there is no, GWAS is very mature. Every literature refers to pretty much the same model. So it should really just be one more of the elements of our battery of things, and almost done in a daily basis, not necessarily daily basis, but in a regular basis. So before I go, this is still running. My computer is a little bit slow today. So while we wait for that, I might be able to answer a couple of questions, Carrie.
Sure. Okay, so a question here from Sanjeev. How critical is it to transform the phenotypic data if the trait distribution is skewed?
It is quite important, because the residuals are assumed to be normally distributed, which also drives into the effects belonging to a normal distribution. And that means if they're done, your p-values are going to have some conflicts because they refer to a different distribution. So it's important. It's an important thing.
And Cesar's wondering if we can share the script in the tutorials to replicate the results. I think most of it's available in the manual. Is that correct?
Yes, most of it is available in the manual. These examples were mostly pulled from the manual, but we'll try to put a link on the recording so you can also download the script if you want to play with it, and some other data sets. Yeah.
I appreciate we're going over time, people. So once again, if you do need to leave, the recording will be provided along with answers to all of your questions.
Okay, so this is exactly what I didn't want to happen. So we have to wait for this to finish. So just to give you an idea of what it comes through it, it will select a number of markers. The markers that are being selected will correspond to both additive and dominance effects. Oh, it seems this is going to be the fast part. So we have 22,000 markers, 1,481 sample. So one thing I want to mention here is that there is a calculation of the heritability for additive effects and a calculation of the heritability for the dominance or dominance ratio in this particular case. And we'll find markers that have a D, which are the markers that are associated with a dominant effect, the 010 coding. Okay, so it finished. It says a few elements about ASReml maybe requiring a few more iterations. Let me just show you the plot. Again, nothing too serious to worry. The variance components. We have the additive component, the dominant component, which is much smaller, the error term. These are the heritabilities I mentioned. We have the heritability of 0.22 for additive, 4 for non-additive, and the join. This is all in the base model. So that means there is some variability to explain on the dominance, but not much and probably not significant. Again, this is an illustration example. And finally, the markers that are being selected. We have a group of markers selected for additive effect, and we have these 5 markers that are being selected for dominant effects with different p-values that are potentially interesting to be looking into. Okay, so that's pretty much it. So I'm going to finish here. Sorry, I'm a few minutes over. But again, thank you for joining us, and hopefully this gives you new ideas or new ways to explore your genomics data. And we are happy to receive any comments, input, requests, questions, etc. from your side. Thank you very much.
Lovely. Thanks so much, Salvador. Appreciation in the comments. That was very well received. I hope you all got a lot out of it. And as we say, we will be sharing this webinar, the recording of the webinar. We will be answering your questions and adding any more information that we think might be helpful. Thank you all so much once again for joining us. And I hope you join us for future webinars, probably not until after Christmas now, I should think. Thank you once again, guys. We're going to close it off now.